The first half of the talk is assuming that you are not an expert on biocrests, or maybe that you've never even heard of biocrests before. And so if you know a whole lot about them, please bear with me. And then the second half of the talk, I'm going to talk through some recently completed and then a whole lot of ongoing and just about to start research involving restoration and biocrest. So what are biocrests? BioCrest is the abbreviated term for the long name biological soil crust. It's the hip way to say it, you guys. And so biocrests are soil surface organisms that intimately associate. So a group of organisms that are living very closely together and associating on the soil surface, and they aggregate the top few millimeters of soil into a coherent layer. You may have heard them called, let's see how this works. Okay, cryptobiotic crust or cryptogamic crust in the past. And these days we all seem to be settling in on biocrest. And I want to make sure that we're clear on how biocrests are different from physical soil crusts, which you may have also seen. Physical soil crusts result just from weathering. It's just soil. There's no organisms there. They have this distinct cracked and platy structure. And so I'll be showing you lots of photos of biocrest, so hopefully this distinction will become more clear as the talk progresses. So biocrests are communities of organisms, I say. So which organisms? So there's mosses. That's how I got to biocrests, because it turns out the mosses I was studying here are in biocrest. And then there are lichen, there are cyanobacteria and algae and other microorganisms, which are even less studied and get lumped into that phrase. And so the amount of any of these four organisms that's in a biocrest community varies across locations and is primarily determined by environmental factors. And so if you've heard of biocrests, you probably think of a location like this in the arid Southwest. Here, it's not as clear as I hoped, but this is a picture in the Mojave Desert. These darker bumps here on the soil surface are all biocrest. And then these two photos are taken from the Colorado Plateau. All the darker places on the soil surface are biocrest. So they're very important in these dry lands. But we also have them here in our semi-arid grasslands and shrublands. This is an insert of probably like an Idaho fescue, and then on the soil surface around it, we have lots of lichen and moss. And so we have a very generalized classification system for the main types of biocrest that occur, and it's mainly separates these types of biocrests based on the organisms that are present and the type of topography they create on the soil surface. And so at the drier end of the spectrum, in hyper-arid and generally easy to disturb locations, we start to see the formation of light algal and fungal biocrest. And in this photo, you can see this lighter area on the soil surface, and that's where the biocrests are forming. Here in the darker area, they haven't started to form. They're not always easy to see, and so one way to know that the biocrests are in this type of location is to take a little bit and do a cross-section, and that's what we see with this insert. And from the cross-section, you can start to see these kind of dangly filaments, and those are fungal filaments, and they're holding together the soil particles. They're starting to form that cohesive layer of soil. In general, these biocrests they keep that basically very flat surface topography. It's not a complex topography. Moving into a slightly less arid location, but still very arid hot deserts, you start to get moss and lichen and cyanobacteria in the biocrest community. But it's still very patchy, it's very dry, and the topography of these biocrests is described as rugose and very patchy. So you can see them every so often, but they're not everywhere, right? Switching from arid deserts to cold deserts, the big change is the freezing temperatures. And so those freezing temperatures, of course, give us frost heaving in the soil. And We get also more lichen and moss in these biocrusts, and we get these pinnacled topographies. They're really quite spectacular, and the pinnacles can actually be up to 15 centimeters tall. And then moving even into less arid locations, when you get into cool deserts, semi-arid grasslands, semi-arid shrublands like where we live, like in the Great Basin, we start to see lichen and moss becoming more present in the BioCrest community. There's a lot more precipitation, so these organisms can occur at greater abundance. And they tend to create this rolling topography on the landscape between the vascular plants. And these are the types of biocrests you would see here, if there wasn't snow covering it right now. So I've talked about how these biocrest communities can vary over a large spectrum of environment, like the large environmental gradient, sorry. But even within a single location, we see a lot of variation in the biocrest communities. So these photos were taken about 1 meter apart on MPG Ranch. So MPG Ranch is a private piece of property about an hour south of Missoula. It's managed for conservation and research. It's a wonderful place to do work, and I've been working there off and on since 2012. And they're way into BioCrest, which is great. And so you can probably just barely see a little moss, the green there. The rest of this is all lichen. And then about 1 meter away, though, the BioCrests look like this. All the green is moss, and there's very little lichen. So within a location, we start to see that the BioCrest community varies. These biocrests have been heavily studied, and so we actually know that there are over 72 species of lichen that occur in these biocrest communities and 25 different bryophyte species. So there's a lot of diversity, even within a single location, tons of diversity across that hydrological spectrum that I mentioned earlier. And so these differences in the biocrest communities, the occurrence of the different biocrest organisms, and the effects they have on soil surface topography all drive how each biocrest community influences their ecosystem. And one of the first and primary ecosystem effects we think about when we think about biocrest is their ability to aggregate soil, to pull together these soil particles and consolidate them, right? And this can be very important in drylands. This is a photo of one of those horrible dust storms you get like in Arizona. The winds come across very disturbed areas. There are no biocrests there holding down the soil surface, and these are clearly very detrimental to human health. They create a vicious cycle where the soil stays unstable. Biocrests cannot establish, vascular plants cannot establish. And so having biocrests really cuts down on the severity of these sorts of storms. The increase in soil stability can also influence soil hydrology in a couple of different ways, so I'll walk you through those. And so here we're going to focus in more on mosses, but all biocrest organisms, being experts at living in dry locations, are able to go more or less dormant when water is not available, right? They don't have roots, they don't have vascular systems, they can't scavenge for water like vascular plants can. So they can go dormant and they can stay like that until rain or precipitation comes back again. Mosses in particular are very amazing at this rehydration part, and they can reabsorb up to 100 times their dry weight in water. They store it in their cells, which balloon out very quickly, and they store water all in these tiny interspaces between the shoots. And so effectively on the landscape, they're like tiny sponges everywhere. And they drastically increase the ability of that landscape to capture and retain moisture during the times when it's available. And then the other way that this increase in soil stability influences hydrology, I think, is best shown with these lichen and cyanobacteria-dominated crusts. And you can just see these pathways that the water is traveling through the biocrust. And just by changing the way the water moves across the soil surface, by creating this new twisty pathway, it slows the water down as it moves across the soil surface, and it increases its opportunity to be absorbed, and it decreases the amount of soil taken away in erosion. And so these positive effects that BioCrest can have on soil stability and soil hydrology can also regulate soil nutrient cycling dynamics. Nutrient cycling dynamics in drylands are almost entirely regulated by water availability, because that's how everything starts to move and change. These same effects will also buffer soil temperatures, and in particularly keeping those temperatures, or the extremes of those temperatures, from being quite as extreme. In addition, by combining all of these ecosystem services, BioCrests are very clearly influencing the establishment and growth of vascular plants. And so this brings us to the point that's my favorite thing about BioCrest, and my favorite area of research right now, is that BioCrests are coexisting with vascular plants. When we think about restoration, I'll be honest, everyone except me thinks about restoring the vascular plant community. People tell me this all the time. What are you going to do for the vascular plants? But the BioCrests are coexisting with these vascular plants. They're influencing their establishment at many different phases. And so it's really important that we understand how they're interacting, and I think we can use this to our benefit. And so I'm going to go through just a few areas where we know that BioCrests in particular influence vascular plant establishment. There's some things I'm not going to have time to get into. So particularly, BioCrests influence plant establishment during this phase of seed capture. So I have all these pictures of amazing seeds with crazy different morphologies, forbs and grasses. And I used to think of these different morphologies as really important for dispersal. And I thought, sure, a deer has to come along, and the seed has this morphology to latch onto the deer and get to a new location, and that's why it grows that way. But it turns out there's more than one reason. And so with this cartoon I have here, we have seeds coming into a hypothetical environment that has bare soil and different kinds of biocrest with different topographies, right? The seeds can land on all these topographies. That's no problem. But once the wind and the rain comes along, many of those seeds will be blown away. But when we have these more complex topographies, like occur with these rolling biocrust or pinnacled biocrust, more of the seeds are captured. And the seed morphology can play a big part in that. Then when we think about plant growth, this positive effect that biocrests have on soil water availability and soil nutrient availability is very important in dryland systems, where the period of time that seeds and seedlings have to establish can be very brief. And so they need to do it really fast. And so the biocrests are extending that period of time that can be beneficial. So not to mention, if they've trapped more seeds, maybe we could have more plants growing there at the same time too. So there's a very clear discrepancy here that I've left out, and maybe you've caught. Like, we see a lot of bare soil, a lot of very disturbed soil, and there's a lot of things growing on it. And that generally it's the exotic invasive plants that we really don't want to be there. That's why we're in this career anyways. And so this brings me to another point, something else that's really cool about BioCrest. BioCrests tend to influence the establishment of native and exotic plants differently. And we see this most strongly with one of our worst invaders, cheatgrass. And so there's been studies on the Colorado Plateau and the Great Basin, and then my own work here in the Intermountain Grasslands have all found that when biocrests are intact, cheatgrass establishment is inhibited. And I found the same thing for spotted knapweed and sulfur cinquefoil. So effectively, healthy biocrest communities can inhibit the establishment of our first, second, and fourth highest impact invaders in this system. Unfortunately, once biocrests are disturbed, the positive ecosystem effects and the ability to keep exotic plants from establishing, those benefits are gone. And biocrests are particularly sensitive to compressive forces, such as those that occur with grazing or off-road vehicular travel. Biocrests can be damaged by fire, and in particular, they're severely damaged when those fires are very intense. And maybe most unfortunately, biocrests are very, very, very slow to recover on their own. And even more, it appears that when biocrusts are disturbed, somehow that disproportionately benefits exotic species. So this is from the same study that I was just telling you about, where I, in the Missoula area, found that intact, healthy biocrest communities limited the establishment of cheatgrass, knapweed, and linaria. But once those biocrest communities were disturbed, we saw that those exotics increased in establishment to 4 times that of natives. We don't understand the exact mechanism behind this, but we think it probably has something to do with that positive effect that biocrest can have on soil moisture and soil nutrients. And this recent study by Ferran Berg kind of makes this distinction clear. I apologize, it's always blurry, but it's just so clear. And so they grew cheatgrass in soil where biocrests had been removed, and they grew cheatgrass in soil where biocrests were disturbed. And this is a very healthy cheatgrass, and that came from the soil where the biocrest had been disturbed. And so there's been a lot of work in the field of community ecology and ecosystem ecology to understand what biocrests are doing, to just generally increase the knowledge of biocrests, make people aware that these tiny little things on the ground everywhere are there and that they're important. But we hear a lot less about biocrests in the field of restoration ecology. And so I've been working with these amazing people to change that. And so all the ideas and research that I'll be sharing was developed in collaboration with Dean Pierson from the Forest Service, Rebecca Durham from MPG Ranch, and Nicole Barger from the University of Colorado. And most of our work is centered on trying to preserve and minimize our impact on biocrests that are already intact and there, they haven't left yet, and then to develop methods for reestablishing biocrests once they're disturbed to reset that ecosystem function that we can really benefit from. So I'll speak on this first point first. Preserving biocrests is pretty cut and dry, right? And I think it mainly comes down to improving our awareness of biocrests. So maybe someday when the snow melts and you're on a walk out here in your grasslands and it's recently rained, take a look a little closer. If you've never noticed, there's actually moss and lichen all over the ground between the vascular plants. And they can really be quite spectacular. And you really notice them after a rain. They're bright green instead of being black. But most of the land we're dealing with is managed. And so whether it's being managed for agriculture or for recreation or for grazing, we really have very little understanding about how the management practices we're using are influencing the biocrests that are there. And so I'll first talk about some work we're doing to understand how herbicides and herbicide surfactants are influencing biocrests. And I'll go ahead and spill the beans and say we have no idea. We have no idea how these chemicals influence biocrest. But what we do know is how they act on vascular plants. And so we started thinking about it when we thought about these differences in their morphology and how they function physiologically. And so let me show you. Here we have a cross-section of a vascular plant leaf, a cross-section of a moss leaf, and a cross-section of a lichen thallus, or the main part of the lichen. So we all probably know that the exterior of a vascular plant leaf has a very waxy cuticle that protects the interior of the leaf. That waxy cuticle is impermeable to water. Water does not enter leaves through the leaf tissue, right? It has this cuticle to keep everything out. And so one of the reasons that they add surfactants to herbicide mixtures is because the surfactants, though they act many different ways, they often break down that cuticle of the leaves, and they then allowing the herbicide to enter the leaf tissue and kill the plant. Moss leaves are 1 cell thick. That's it. There's no waxy cuticle. There's nothing else to protect it. They get water through the simple process of diffusion. They have very little control over it. Lichen thalli are a little more complicated, but if you remember, lichen form as the symbiosis between multiple organisms, right? We've got a fungi and a cyanobacteria, or maybe an algae, maybe multiple fungi. And so with lichen, we do not have, again, a waxy cuticle. We just have this layer of fungal cells at the top, and then a layer of either algal or cyanobacteria cells beneath. So there's even multiple organisms that could be influenced, and we don't know how they're being influenced at all. And so the first goal is to kind of get an understanding of what's happening here. How are herbicides and surfactants influencing these organisms? But it could be that the timing of herbicide application might have different effects on moss and lichen. So this is something else we're thinking about. Maybe if the herbicides are applied when the biocrusts are dry, they may not hurt them at all. But maybe it would hurt them a lot more if they were applied when the biocrusts were wet. So we're starting to play with that timing of application. Additionally, we know there's many different kinds of herbicides, many kinds of surfactants. They're often applied in cocktails to be most effective, and so we're testing many different kinds of herbicides. And then, like I said, we have a lot of diversity in these BioCrest communities, and so we have to test the different herbicides at the different times on the different BioCrest organisms. So there's many ways this could work out. And right now, we're just kind of throwing darts. And so this is the first study that we've done. This is just a list of the herbicides and their surfactant, the one surfactant that we've tested. And we're testing them by applying them to biocrests dominated by moss or lichen, because that's primarily what we have in our grasslands. And we do expect that they might respond differently. And so right now, I have the first data from this coming in, so I don't have any results for you yet. But unfortunately, it did look like as soon as the herbicides were applied, the mosses all turned bright red. But mosses are pretty burly, and I'll talk about how burly they are a little later in the talk. So there's a good chance they just grew right back. And so we'll see, and I'm excited maybe someday to be able to share that with you. And then another area of research we're interested in is the reseeding efforts we do in restoration. So I talked a little and kind of brushed over, to be honest, that influence that biocrests have on native plants. Because there's so many native plants, there's so many biocrests, we don't know a whole lot, to be honest, in any one system. It does seem to be that different functional groups might be influenced differently by biocrests. There's also some species-specific effects. But as a restoration practitioner, you could save a lot of money and really benefit from knowing if you threw your seeds on the moss or on the lichen or on the bare soil, which one has the better chance of growing? Because that's the ultimate goal. And so in another recent study that we just started, we added the seeds of just this small subset of grasses and forbs, again, to biocrests dominated by moss, lichen, or to bare soil, just to see where we get the higher recruitment. And so I'm excited to see what happens there. But with the previous study and with this one, there are so many variables. And it gets kind of hard to know where to start and where to go next, right? I mean, sure, if a whole bunch of people start studying this, we could really cross some things off our list. But I got to recruit some people, I guess. And so what we're trying to do is apply a little more ecological theory to our studies and integrate that in a little and see if we can move forward a little faster. And so many of you may have heard of community assembly theory. Community assembly theory posits that the composition and abundance of species within a community is influenced or determined by a series of hierarchical filters. And these filters either allow or impede the passage of a species into a system based on its traits. I'm going to explain all of this. And so if we think of each of these shapes as a different species, not all of these can fit through this filter. And so we can assume maybe this filter is dispersal, right? Maybe one of these seeds needs a certain organism for dispersal. If that organism is not there, that species cannot pass into this community. The second filter could be temperature. If the species may be able to get there, but it may not be able to survive the temperatures that are already in that community. And so we're really interested in understanding how the filters of precipitation and biocrest, which we know to be important filters in our dryland systems, are influencing the composition of our native plant communities. And so I'm doing, this research was just funded, and it's going in the ground, just went in the ground in Utah. This is what I've been doing the last month. And I'm about to put it in the ground in Missoula. It better not snow there, or I'm gonna have a really hard time. But, and what we've done is we've installed a whole bunch of different rainout shelters. And these will get covered in the spring, because the goal is to block out the ambient rain during the period of time when the seeds are germinating and establishing. And I'll go into this more. And then under these shelters, I've put in lots of pots, and I harvested these BioCrest cookies on the landscape. And then I put the BioCrest cookies in a pot, and I either leave them intact or this one is disturbed. And then on top of those BioCrest, I put the seeds of a single species. And I've done this for over 25 species. And so we'll get a good idea from this of which plants are able to survive and establish on biocrests that are intact and disturbed, under normal precipitation regimes, and under precipitation regimes that are slightly higher than normal. And then we'll combine that data with trait data that we're going to collect on these seedlings. So the fact is that lots of seeds germinate. Germination doesn't seem to be the major problem. But the majority of seeds that germinate die during these very early life stages. And if you think about it, we might have a precipitation event, and then it might not rain again for weeks. And so for that seed that germinates, for that then seedling to live, it has to get its roots deep, deep in the ground to an alternate water reservoir. And that can be very challenging, if not impossible. And so that's why we have such high mortality. And so we're really interested in looking at some traits that describe plants during this early, very vulnerable time period. And so some of these traits we're interested in describe like seed vigor, so looking at seed mass and seed germinability. And then we're interested in seed phenology traits, like the time to germination and the range of germination timing throughout a season. That could be very beneficial. And then, like I said, if the seedling can get its roots deep in the ground really quick, that would be great for that plant. But in the same ways, if it grew up and was able to make that true leaf faster, there's only so many nutrients in that seed reserve. And so the sooner it can make a true leaf, the sooner it can photosynthesize and make its own energy. And so that could benefit that species. And so we'll be combining the data from the field experiments where we see which species survive, and then hopefully we can start to see maybe why those species are surviving. And we want to use this information to basically do community reassembly and restoration and create seed mixtures of species that have these traits that might make them more likely to survive, both on areas where biocrests are disturbed, areas where biocrests are intact, maybe these mixtures of species that could survive both high and low rain events to kind of hedge our bets. And so this is something that we're very excited to see the results of next spring. And so I've talked through these first three experiments, and then now I want to move into things we're doing to understand how to reestablish biocrest once they're disturbed. So this is where I said mosses are burly, and let me explain why. And really this is the same for all of these biocrest organisms. Biocrest organisms are renowned for their ability to regenerate. And so if you take this moss leaf and you dry it and you crumble it into tiny bits and you spread that out, every single cell can regrow itself. It won't regrow a leaf, it'll regrow a moss shoot. I just had to clarify. I don't want you walking out of here with incorrect information. But that's a very amazing thing, and it makes it really easy for us to get the propagules we need for restoration. We just need to harvest biocrust, or ideally salvage biocrust, and then ground those up and spread them around, right? And salvaging biocrest is great when it happens. I do know a lot of people that are managing to do that and work with like construction crews or building crews and get in there and salvage biocrest. But generally, there's not as much there to salvage as we need. And so if I go out instead and harvest biocrest, then I'm creating another disturbance. So the goal is to figure out some healthy balance here, right? And either harvesting a minimal amount or salvaging a huge amount. And so we want to minimize the amount of inoculant we're using in these reestablishment experiments. And then the BioCrest fragments themselves are very lightweight. Think like the weight of like a dog hair. And so if the wind comes along, they can blow them away. So another goal is to reduce the loss of these fragments when we have them. And so we set up a really big experiment in a common garden setting at the MPG ranch that I mentioned before in 2012. And we added BioCrest fragments, and we found that we could reestablish BioCrest in these plots using only 12 grams of BioCrest fragments per meter square. And so that's like the amount of tea you might use for a pot of tea. So not a whole lot. If you use more BioCrest fragments, it does establish faster, but we could do it even with this minimal amount. And so we were very excited by that. And we also found that more of those fragments stayed in place when we added jute ground cloth. The BioCrest seemed to work really well with this jute ground cloth. And these figures, it hardly matters what they actually say, but this is moss BioCrest cover with no jute. It's practically zero. It just all washes away. And then with jute, whether you apply the inoculant below or on top of it, it doesn't really matter. You get drastically higher BioCrest cover when you add jute ground cloth. And we also found, which this was a little surprising, that the reestablished BioCrest decreased soil nitrogen. And why that could be helpful is not just immediately intuitive, maybe, but if you think about when you have an area that's been recently disturbed, you often have this pulse of nitrogen and this increase of nutrient availability. And that really benefits exotic plants, invasive plants, and helps them establish. And native plants don't do quite as well with that. And so being able to reduce these increased pulses and pools of nitrogen could actually facilitate native plant establishment. So we hope. And so that's a very exciting thing. But like I said, if we're not salvaging BioCrest, then we'd be creating another disturbance by harvesting BioCrest. And so there's actually a lot of labs working right now on BioCrest cultivation. And they're primarily doing it in greenhouses. It turns out these organisms can do really well in drylands, but if you pamper them, they grow really fast. And so they're able to grow the BioCrest much faster. That experiment I just showed you, that's what I wanted to tell you. That took 4 years. So it was very slow to regain cover. Someone had to hand weed it. That does not help us at all, really, with restoration in the real world, right? And so we need something that happens much faster. And so what these folks are able to do is grow enough BioCrest that we can use as inoculant, so we don't have those additional disturbances. Grow enough BioCrest that we can use to add a lot of inoculant so that they can grow faster. And then they're also experimenting with some extra cool ideas. So this is my favorite. So in the greenhouse, there's these shelves, and each shelf is covered in burlap, and then the burlap is covered in BioCrest fragments. And then they kind of rotate around, and so the BioCrests get access to the sun. And so you're able to fit a ton of burlap sheets within a greenhouse that way and grow a lot of BioCrests. Still, and it's not like we have rolls and rolls of turf, BioCrest turf, that we can roll out and cover hundreds of acres, which is more the level of restoration we need to do. But we have been starting to think about using these in targeted areas that are very difficult to get anything to establish in, but we really don't want, say, cheatgrass to come in there, because then we're not going to get rid of it. And so if we can lay down these burlap mats, the BioCrests seem to survive really well. They seem to be able to spread out from the burlap mats really well. And then maybe we'd have time to plant in some vascular plants and move that little area forward. And so this is an area, like I said, that's receiving a lot of interest right now, and I expect it. I was actually, I'll admit, I was very, very skeptical when I started doing the basic restoration of BioCrests, because you see the scale of the disturbance that needs to be dealt with sometimes. But I'm starting to see the light and starting to see that there's enough people working on it that I think that there'll be some really cool tools to use in the very near future. And so I talked really fast. I'm sorry. And I talked through about 5 different things that are ongoing in my research group and with these folks. And I want to acknowledge quickly our funding sources, the National Science Foundation and MPG Ranch, but I did leave plenty of time to answer any questions. So maybe you have some.
Thank you.
Yeah.